All right, there's three major mechanisms of active transport. There's the coupled transporter, right? Three major mechanisms of active transport. There's the coupled transporter, which we could say is like secondary active transport. Sometimes we call it secondary active transport, okay? Coupled transport. And who's our favorite example of that? Not the glutes. The sodium glucose transporter. The sodium glucose transporter. That's a SYMPort, remember? So there's three major types of active transport, okay? A secondary active transport, such as the sodium glucose transporter, right? We've also talked about other mechanisms of active transport that instead of using sodium, the sodium gradient, that can use the proton gradient, correct? So if you go ahead and take a look at what we talked about for in chapter 13 and 14, we also have some SYMPorts that actually allow what? That actually allow hydrogen protons, okay, to come across the what? The inner membrane of the mitochondria in order to bring something in against its what? Its electrical chemical gradient. All right, so we have coupled transporters. And then, of course, what we have is what? We have also the sodium, the ATP-driven pumps, such as the sodium-potassium pump, correct? Right? And then we even have in nature, we have some light-driven pumps, okay? Light-driven pumps like bacteria. Like bacteriorhodopsin, okay? That actually is found in bacteria, in certain bacteria, photosynthetic bacteria that actually can use what? Solar energy to pump hydrogen protons, okay, across the plasma membrane, okay, light-driven pumps. So those are the three major mechanisms of active transport. Obviously, these two are the most important to us, but there are some what? Photosynthetic organisms, such as the photosynthetic bacteria, the purple bacteria, actually, the bacteria, okay, that actually have what? A membrane protein that can pump something, based upon light. Bacteriorhodopsin actually does that by what? Pumping protons across the membrane in the presence of light. So, like bacteriorhodopsin, I even spelt it right? Good. Okay, the sodium potassium pump is not the only ATP-driven pump. Another very famous ATP-driven pump would be the ATP-driven calcium pumps. The ATP-driven calcium pumps that are found, for example, in the endoplasmic reticulum, okay, and also plasma membrane of animal cells, okay? And of course, these ATP-driven calcium pumps are very important at keeping calcium levels in the cytosol low. Okay, so what we want to do is the goal of these guys, the goal of these guys is to keep calcium levels low in the cytosol. Okay, so where do we find these calcium-driven, ATP-driven calcium pumps? We can find them in the plasma membrane, and we could also find it in the endoplasmic reticulum, particularly in muscle tissue. The endoplasmic reticulum in muscle tissue, in skeletal muscle tissue, is called the sarcoplasmic reticulum. And as we know, okay, anytime there's going to be a release of calcium into the cytosol of a skeletal muscle, what's going to happen? It's going to initiate muscle contraction. So once calcium comes out to initiate muscle contraction to take place, we have to pump the calcium out of the cytosol and store it back in the lumen of the endoplasmic reticulum, or better known sarcoplasmic reticulum, skeletal muscle cell. All right, so there's the calcium pumps that are ATP driven as well. So you could take a look at that. And let's just talk a little bit about cytosolic calcium being an important signaling agent. Calcium pumps are super important for what? That's supposed to be for for keeping cytosolic calcium levels low. Besides the role calcium plays in muscle contraction, we know that releasing cytosolic calcium or increasing cytosolic calcium levels affects gene regulation, affects vesicular exocytosis, such as at the end of the axon of a neuron, right? Can influence cell growth and division, okay? And can be involved in signaling transduction pathways, such as the CAM pathway and also PKC. Also can be involved in calcium regulated chaperone proteins like calnexin and calcium dependent proteases like calpase. So in other words, calcium ions are a super important signaling molecule, okay, in the cytosol of cells. And so therefore we have to be very careful about making sure that we can regulate calcium levels, that when calcium comes out in the cytosol, it does its thing, and then very quickly we bring back down the calcium levels, okay, in the cell. And we do that by a number of different mechanisms, including the ATP driven calcium pumps that can be found in the plasma membrane of animal cells and in the endoplasmic reticulum as the two most important areas where we find that. Lysosomes have an ATP-driven proton pump. ATP-driven proton pump. All right, so we're not only doing sodium-potassium pump, not only talking about an ATP-driven calcium pump, but we also have ATP-driven proton pumps. Okay, and lysosomes have that. And why do we have that over here? So we could turn around here and we say that we're going to use cytosolic ATP hydrolysis in order to pump hydrogen protons into a lysosome. And why are protons actively pumped into the lumen of a lysosome? What's the major role of a lysosome? Lysosome has all these digestive enzymes. Okay? And the optimum pH for these digestive enzymes are usually around a pH of about 5. And so even though the pH here is around 7 in the cytosol, the pH inside of a lysosome is going to be what? Around 5. So the way that we maintain that difference in pH concentration is by using ATP energy to keep pumping in hydrogen protons into the lumen of the lysosome. So just here they're showing you what? They're showing you an ATP-driven proton pump that's found in the membrane of the lysosome, which pumps hydrogen protons into the lumen of the lysosome. It brings down the pH so that the digestive enzymes work at their maximum efficiency. Okay, here we are talking about using a sodium-driven Symport. Okay, sodium-driven Symport to let sodium come in down its electrical chemical gradient and bring whatever that solute is, whether it's glucose or something else, to bring that in against its concentration gradient. And then here we have the sodium-potassium pump. So in an animal cell, we have these different types of membrane proteins that are actually using different types of energy, whether it's ATP energy primarily, or whether or not it's secondary active transport, like the sodium glucose transporters. Jeez. Right, the solute might be glucose. Right, glucose. So different locations for these different types of pumps in animal cells, such as our cells, correct? Plant cells have an ATP-driven proton pump in their plasma membrane. Remember, they don't have a sodium-potassium pump, and so therefore the plasma membrane of plant cells and bacterial cells is not a sodium gradient, but it's actually a proton gradient. Okay, so animal cells use a sodium gradient as their what? As their battery, as their cause of their membrane potential across their plasma membrane. But plant cells and bacterial cells have a proton gradient. Instead, they do not even have a sodium potassium pump in their plasma membrane. So the membrane potential that they have across the plasma membrane is based upon a what? An electrical chemical what gradient of protons. Right? So therefore they have to have an ATP driven proton pump over here. And why do they do that? Then they have a what? They pump out the hydrogen protons, cause a membrane potential, cause a proton gradient to form across the plasma membrane. And then instead of using a sodium driven symport to bring in solutes against the concentration, they bring in solutes based upon what? The proton driven symports. Okay. And of course, they don't have a lysosome. Plant cells don't really have a lysosome. The so-called lysosomal function of the plant cell basically is accomplished through the what? Central vacuole. So the central vacuole also has a more acidic pH, okay, and also has somewhat digestive or lysosomal-like activities because plant cells really don't have their own lysosomes like animal cells do. Okay. All right, all right. Urobilin and digitalis work by partially inhibiting the actions of the sodium-potassium pump in the plasma membrane of cardiac cells. Okay, so digitalis drugs and urobilin, which is another chemical, works by inhibiting the sodium-potassium pump. This is potentially dangerous, correct? To be exposed to these drugs, and digitalis is a pharmaceutical drug, correct? However, at therapeutic doses, it can be very helpful to a person who is actually undergoing heart failure to be given a digitalis drug. And the way that the digitalis drug works is that it inhibits what? Partially inhibits. Can't completely inhibit, you kill the person. Partially inhibits the action of the sodium potassium pump. Result of this, if the sodium potassium pump is not working that well, okay, it means that the membrane potential is begun to become less. Cardiac muscle cells usually do have a resting membrane potential of around minus 90 millivolts, more so than a neuron. If the sodium potassium pump is not working as well, that means instead of the membrane potential being like minus 90, it might be down at minus 70 or minus 60, a little bit less than that. Okay. And when that takes place in a cardiac muscle cell, it makes it harder for the cardiac muscle cell to pump out cytosolic calcium. Because unlike the skeletal muscle, who gets really all of its cytosolic calcium from the sarcoplasm reticulum after an action potential, cardiac muscle cells get their calcium levels, their cytosolic calcium, from both their sarcoplasm reticulum and also from extracellular calcium. So the extracellular calcium, once it comes in to initiate a cardiac muscle contraction, eventually you have to have relaxation and you have to pump the what? You have to pump or somehow get the calcium out of the cell. And so therefore, if you actually, one of the things that the cardiac muscle plasma membrane uses to get the calcium out of the cell is that they use this sodium calcium antiport. Okay. So since they actually, a lot of their calcium from the extracellular fluid, they have to have more than just a calcium ATP-driven pump to get the calcium out. They use a sodium calcium antiport. And the only reason that the sodium wants to come in is because the membrane potential is so nice and negative. But now that it's less negative, there's going to be less of a desire for the sodium to want to come in, and that means that there'd be more of a tendency of the calcium to have trouble getting out of the cytosol. The effect of that in a cardiac muscle cell, okay, for a person who has heart failure, is that it increases the strength of the beat. It might actually decrease the pulse rate, but it definitely increases the strength and improves the cardiac output for the people who have heart failure to actually have what? More calcium present. It makes it a stronger contraction of the heart muscle. So in other words, it works therapeutically. However, it would be very dangerous to have an overdose of these drugs, which is always a big problem, especially with elderly people. There's many reasons. They're on multiple different types of drugs. Some of them actually cause an increase in the half-life, or prevents the quick breakdown of the digitalis drugs, and increases the accumulation of the drug in their system to potentially toxic levels. And that would end up being very dangerous, because if you totally prevent if you totally really decrease the sodium potassium pump, okay, that's going to end up causing what? All sorts of electrolyte changes, and that's going to end up causing even maybe the heart muscle to suddenly just contract and not even relax. Okay, so you can get all sorts of arrhythmias and atrial fibrillation as a result of too much of calcium inside the cell. All right, so let's see, getting close to the end of what I want to talk about. We just want to talk about the parts of a neuron. Okay, so people who already had a lot of other biology courses should know, first of all, that there are 3 different types of neurons, 3 basic types of neurons. We have the sensory neurons, which actually send information, okay, to the central nervous system, right? The sensory neurons send the information to the sensory nervous system. Okay, the motor neurons, which send out a message from the central nervous system to the peripherals and to the muscle tissue, correct? And then we have the interneurons. The interneurons would be the neurons that are communicating within the central nervous system. So those are the 3 different types of neurons. And then when we talk about a neuron, we say a neuron is made up of 3 major parts, right? We would say the dendrites, which is the incoming, okay? So in other words, if one neuron is going to communicate with another through a synapse, okay, the information is going to come in through the dendrites, right? And then, of course, the cell body is the main part of the neuron, which contains the nucleus of the cell, correct? And then, of course, we also know that that neurons can have these axons. Okay, and the axon is what's going to generate, or where with having the action potential that allows one neuron to then communicate with another neuron, correct? The axons are very interesting. In fact, the whole shape of a neuron is so bizarrely different than most of our cells, correct? Because some axons, especially in the interneurons, can be less than 1 millimeter in length, okay, especially in the interneurons. Whereas when we talk about some of the axons, especially of our motor neurons, some of them can be more than 1 meter in length. That's 3 feet. Okay, so one cell can have an axon that actually is more than 1 meter in length. That would be particularly true with some of our motor neurons, correct? All right, no matter what the message is supposed to be, the form of the signal is always the same. Okay, so let's talk about how we could have changes in the membrane potential, which can open up voltage-gated ion channels, okay, along the axon and then even at the axon terminal. So let's see whether we can do that.
The fundamental task of a nerve cell is to receive, conduct, and transmit signals. Neurons propagate signals in the form of action potentials, which can travel great distances along an axon without weakening. To transmit an action potential over such a distance without weakening requires that the signal is continuously reamplified along the way. The central molecular players in this process are the voltage-gated sodium channels, which undergo a cycle of finely choreographed conformational changes. When an action potential passes, sodium channels open in response to membrane depolarization. Sodium ions rush into the axon, further depolarizing its membrane. Within a fraction of a thousandth of a second, however, the sodium channels switch to a new inactivated state in which they are closed, but now also refractory to reopening. In this way, the membrane potential can recover quickly after an action potential has passed. The sodium channels then reconvert to the closed state, ready to be opened again when the next action potential is encountered. Let's examine the changing state of the sodium channel during an action potential. When no stimulus is present, the sodium channels remain closed, and the electrical potential measured across the membrane remains constant. However, if a depolarizing stimulus is applied by a brief pulse of electric current, the membrane will start to depolarize away from the resting potential of about -80 millivolts. Some of the sodium channels will open, permitting sodium to enter the axon along their concentration gradient. If the depolarization is sufficient, even more sodium channels open, and the membrane potential rapidly approaches the equilibrium potential for sodium, about +40 millivolts. At this point, the sodium channels close, adopting the inactive conformation, where the channel is unable to open again even though the membrane potential is still depolarized. The sodium channels will remain in this inactivated state until a few milliseconds after the membrane potential returns to its initial negative value. The action potential is propagated along the length of the axon in only one direction. By examining the membrane potential and state of the sodium channels along a length of the axon, we can see why this is so. As a depolarizing stimulus reaches our section of the membrane, sodium channels open and current flows into the axon. This in turn depolarizes adjacent sections of the membrane, causing adjacent sodium channels to open, and the action potential is thus propagated along the axon. Sodium channel inactivation prevents the depolarization from spreading backward along the axon.
So that's actually really important about in the axon, the three conformations of the voltage-gated sodium channels in these axons, that there's three conformations. So that you would start with what? Closed. You would start with closed. And of course, this is the normal membrane potential, right? So you would start with the sodium channels being closed. And then what? An action potential could suddenly open up the channels, allowing what to come in? Sodium to come in. And if sodium comes in, it's going to depolarize the membrane. And then instead of it just going back to closed, instead of it just going right back to closed, it moves very interestingly into a third conformation first. Okay, it moves to an inactive conformation, one that is closed but is unable to be reopened for a certain period of time. Okay, and that's going to be necessary if the membrane is going to have an opportunity to repolarize. Okay, and it's also going to be necessary to ensure the propagation of the action potential only going forward one way and not trying to go backwards. All right, so the importance of having not just an open and closed state, but to having three states to the voltage-gated sodium channels that are in the membrane of the axon. Okay, and then this is just pretty much the same picture of what we showed before. So what two factors are responsible for repolarization? So we know that we're going to depolarize, right? So here we are, we're depolarizing here, correct? Because we know that what? Sodium is rushing in. Sodium rushes in. Okay, and when sodium rushes in, we're going to go from what? -70 millivolts to -30 to 0 to +20 to +30 to +40, maybe all the way up to +50, right? Very close to +50. What we said before was the theoretical membrane potential equilibrium, right, for sodium, correct? Right? And then, of course, what's happening though, instead of going then from the open state back to the closed, we then go to this other state called inactive state. And what happens during the inactive state? What's happening during the inactive state? See, what's going on here? The membrane potential is back. So now you're positive 40. Now you're positive what? Positive 10. Now you're 0, minus 20, minus what? Minus 40, minus 50, okay? And then finally what happens? You go to your closed situation. So what two factors are responsible for the repolarization? How does it repolarize? And the two factors that are responsible would be the sudden opening of the what? The potassium gated channels. Because once you actually suddenly depolarize, when you depolarize, suddenly now that's going to open up the potassium gated channels. And while potassium normally would not really care about leaving the cell, suddenly now if the membrane potential is positive 40, then the potassium inside the cell go, hey, what am I doing in here? I'm going to run out. Okay, so to allow the potassium now to run out is going to what? Send out positive charges in the cell. And then ultimately you have to come back to like an equilibrium of your concentration. So who's going to be responsible then for pumping out the sodium that came in and bringing back the potassium, the sodium potassium